Back to Fascia Story Time with Dr. Kate Oland, where I'm reading to you from Anatomy Trains by Thomas Myers. We are reading from Appendix 1 of Fascial Reader. Today I'm going to continue on, and we are in the section titled Fascial Components. And we'll start with how to build a body. If we take on the thought experiment of building a human body out of materials that could be bought in a local hardware store or builder's supply, what would we need to get? We will imagine that we already have the computer to run it and that we have already obtained little servo motors for the muscles. But what would we need to buy to build an actual working model of the body's structure? In other words, what kind of structural materials can connective tissue cells fashion? You might suggest wood or plastic pipe for the bones, silicone or plastic of some sort for the joint cartilage, spinal discs, and heart valves. We would fill our cart with string, rope, and wire of all kinds. Hinges, rubber tubing, cotton wool to pack empty places, cling wrap, and plastic bags of all sizes to seal things off. Oil and grease to lubricate moving surfaces, glass for the lens of the eye, ceramic for the teeth, lots of cloth, plastic sacks, filters, and sponges of various kinds. And where would we be without Velcro and duct tape? The list could go on, but the point is made. Connective tissue cells make biological correlates of all these materials and more by playing creatively with cell function and the three elements of the ECM: the available fluid, the tough fiber matrix, and the viscous ground substance. The fibers and ground substance, as we shall see, actually form a continuous spectrum of building materials. But the distinction between the two, non-water-soluble collagen fiber and hydrophilic proteoglycans, is commonly used. The ECM, as we will learn in the section on biotensegrity, is actually continuous with the intracellular matrix as well. But for now, the distinction between what is outside the cell and what is inside is useful. Now we will discuss fascial elements. The list of connective tissue elements is short, given that we are not going to explore the chemistry of its many minor variations. So we have cells that secrete the intercellular materials, namely tough fibers of collagen and elastin, and gluey ground substance into the body's interstitial fluid milieu. Let's talk about the cells that are going to be important here. First, we'll talk about fibroblasts. Connective tissue as a whole has a wide variety of cell types, but many of these are the body's wanderers. Blood cells, red and white. Connective tissues also include mast cells, pigment cells, glial cells, fat cells, lymphatic cells, and blood-making cells in the bone marrow. We are specifically interested in the cells which create and maintain the ECM. They are more limited in category, consisting primarily of fibroblasts. The fibroblast family is nevertheless diverse, and cells can convert from one type to another, though the limits of that interconversion are not yet fully delineated. Osteocytes are bone cells, and chondrocytes are cartilage cells. Fat cells and even smooth muscle cells can derive from fibroblasts, which are pluripotential, like the mesenchymal stem cells from which they themselves derive. The cell type and function can be converted by both chemical agents and, because the cells are velcroed with ligands to the ECM, mechanical changes in the local ECM. Fibroblasts are thus highly generalized and versatile cells which crawl through the matrix, laying down new matrix where loads create more piezoelectric charge, and cleaning up old and molecularly frayed collagen with proteolytic enzymes. The fibroblasts are both the planters and the pruners of our fascial garden. An interesting attribute of fibroblasts is that when the matrix surrounding them is loose and lax, the cells extend processes into the matrix, making the cell bodies small and using the process to monitor local tissues and contact other fibroblasts. As the surrounding matrix is loaded, pulled on, subjected to tension, then the fibroblasts pull in these processes. The cytoplasm gathers in the body, and the gears start whirring to produce more matrix to meet the new load. These proteogenic gears grind slowly, so that a momentary new load on the matrix will not produce more fascia, but a sustained or repetitive load will. This interconvertibility makes the precise classification of this large family of cells problematic. As we can see, bone that has 3 types of cells, one to build new bone, osteoblast, one to eat old bone, osteoclast, and one to maintain it, osteocyte. Cartilage has only one kind of cell, the chondrocyte. In the fascia itself, we have the fibroblasts, which can convert to myofibroblasts, which are halfway between fibroblasts and smooth muscle cells. That's pretty interesting. So we're going to continue on and talk about myofibroblasts. Myofibroblasts. One of the conversions fibroblasts can make is to organize more actin fibers within the cell to become a myofibroblast. This allows large sheets of fascia to be contractile, the only instance we know of active contraction within the fascia. Let us hasten to add that this mechanism works only under limited and interesting circumstances. Wow, I didn't realize how long this section is. I hope you're in it to win it, guys. Given that all cells contain at least some actin and are therefore able to exert some pull, and given that all the cells are velcroed into the ECM with adhesive molecules like the integrins, all cells pull on the matrix. Myofibroblasts, however, are capable of exerting clinically significant contractile force, enough, for instance, to influence low back stability. Myofibroblasts represent a middle ground between a smooth muscle cell, commonly found in viscera at the end of an autonomic motor nerve, and the traditional fibroblast, the cell that primarily builds and maintains the collagenous matrix. Since both smooth muscle cells and fibroblasts develop from the same mesodermal primordium, it comes as little surprise that the body might find some use for the transitional cell between the two. But some surprising characteristics of these cells kept them from being recognized earlier. Apparently, evolution found variable uses for such a cell, as myofibroblasts have several major phenotypes, from slightly modified fibroblasts to nearly typical smooth muscle cells. Chronic contraction of myofibroblasts plays a role in chronic contractures such as Dupuytren's contracture of the palmar fascia, fibromatosis of the plantar surface, or adhesive capsulitis in the shoulder. Myofibroblasts are clearly very active during wound healing and scar formation, helping to draw together the gap in the metamembrane and build new tissue. To be brief, we will let the reader follow the references for these possibly intriguing roles in body pathology so that we can hew closely to our stated goal of describing how fascia works normally. It is now clear that myofibroblasts occur in healthy fascia, and in fascial sheets in particular, such as the lumbar fascia, fascia lata, crural fascia, and plantar fascia. They've also been found in ligaments, the menisci, tendons, and organ capsules. The density of these cells may vary positively with physical activity and exercise, but in any case, the density is highly variable in different parts of the body and among people. Myofibroblasts contract in a spiral motion like smooth muscle cells, not the linear motion of the skeletal muscle fibers. Therefore, they pull in on the whole sheet of fascia, say the crural fascia of your leg when you are on a long plane trip, squeezing the fluid away from your feet back into the venous system. This also pre-stresses the fascial sheet, which allows it to take a larger load and retain its stiffness. Myofibroblasts tighten up the system, turning it from a Nerf ball to a Serpent ball. One very surprising aspect of these cells is that, unlike every other muscle cell in the body, smooth or striated, they are not stimulated to contract via the usual neural end plane. Therefore, they are beyond the reach of conscious control or even unconscious control as we would normally understand it. The factors that induce the long duration, low energy contraction of these cells are sustained mechanical tension going through the tissues in question, and specific cytokines and other pharmacological agents such as nitric oxide, which relaxes myofibroblasts, and histamine and oxytocin, which stimulate contraction. Unexpectedly, neither norepinephrine or acetylcholine, which are neurotransmitters commonly used to contract the muscle, nor angiotensin or caffeine, which are calcium channel blockers, have any effect on these myofibroblasts. Many myofibroblasts are located near capillary vessels. The better to be in contact with these chemical agents. The contraction, when it occurs, comes on very slowly compared to any muscle contraction, building over 20 to 30 minutes and sustaining for more than an hour before slowly subsiding. Based on the in vitro studies to date, this is not a quick reaction system, but rather one built for more sustained loads, acting as slowly as it does under fluid chemical stimulation rather than neural. One aspect of the fluid environment is, of course, the pH. And a lower acidic pH in the matrix tends to increase the contractility of these myofibroblasts. Therefore, activities that produce pH changes in the internal milieu, such as breathing pattern disorder, emotional distress, or acid-producing foods, could induce a general stiffening in the fascial body. Here ends the brief foray into chemistry, which is so well covered elsewhere. Myofibroblasts also induce contraction through the matrix in response to mechanical loading, as would be expected. With the slow response of these cells, it takes 15 to 30 minutes or more before the fascia in question gets more tense and stiff. This stiffness is a result of the myofibroblasts pulling on the collagen matrix and crimping it. Regular fibroblast cells are incapable of mounting the degree of tension or forming the kinds of intracellular and extracellular bonds necessary to pull significantly on the ECM. Under mechanical stress, however, certain fibroblasts will differentiate into a proto-myofibroblast, which builds more actin fibers and connects them to the focal adhesion molecules near the cell surface. Further mechanical and chemical stimulation can result in full differentiation of the myofibroblast, characterized by a complete set of connections among the fibers and glycoproteins of the ECM through the myofibroblast membrane into the actin fibers connected with the cytoskeleton. The contraction produced by these cells, which often arrange themselves in a linear syncytia, as muscle cells also do, like boxcars on a train, can generate stiffening or shortening of large areas in the sheets of fascia where they often reside, as well as laying down additional reinforcing fascia. This discovery, though still in its early stages in terms of research, has implications concerning the body's ability to adjust the fascial webbing. This form of pre-stress, a middle ground between the immediate contraction of voluntary muscle and the fiber creating remodeling shown by the pure fibroblast, can prepare the body for greater loads or facilitate transfer of loads from one fascia to another. In terms of the responsiveness of the myofascial system, we see a spectrum of contractile ability. The near instant adjustment of the skeletal muscle, the more generalized spiral contraction of the smooth muscle, the myofibroblast sustained pre-stress on the larger sheets, and the more passive but still attentive fibroblasts. Given how these myofibroblasts can be stimulated by mechanical loading or by fluid chemical agents, we can also discern in the system the dance among the neural, vascular, and fibrous web that goes into making what we have here termed spatial medicine, how the body senses and adapts to changes of shape caused by internal or external forces. Wow. It's a lot of information to process. So, enjoy the process, and until next time, happy healing.